After the age of 60, something starts happening inside the back of your eye that most people never know about until the damage is already done. It doesn't begin with blurring. It doesn't begin with pain. It begins in a layer of cells so thin it's measured in microns, a layer called the retinal pigment epithelium. And what happens there, silently, every single day, determines whether you will be reading a book at 75 or struggling to recognize faces across a room. Here's what that process looks like. Your photoreceptor cells, the cells responsible for capturing light and converting it into the images your brain sees, are among the most metabolically active cells in the entire human body. They consume more oxygen per unit of tissue than almost any other cell you have, including brain cells. That consumption generates oxidative byproducts constantly. Under normal conditions, a thin layer of cells directly behind your retina handles the cleanup. It recycles the waste. It regenerates the photopigments your eyes need to form clear images. It maintains the blood supply that keeps every photoreceptor alive. After 60, the rate at which that cleanup system works begins to fall behind. Debris accumulates. The tiny blood vessels that feed the outer retina start to narrow. And because those blood vessels are some of the smallest in your body, measuring just a few micrometers in diameter, they are extraordinarily sensitive to the same oxidative damage that is accumulating in the cells they are supposed to feed. The cells get less oxygen. They get fewer nutrients. They begin to degenerate. This is not a condition that develops overnight. It is a process that unfolds over years, often before a single symptom appears. And that is why what you eat this morning, and every morning, matters in a way that most people don't fully understand until an eye doctor is already showing them the damage on a screen. I'm a clinical specialist with over 2 decades of experience working with patients over 60 on exactly this type of age-related retinal decline. What I want to share with you today isn't opinion. It is the mechanism, the biology, and the specific foods that the evidence shows can meaningfully support the structures inside your eye that are under the most pressure as you age. There are 5 of them, and by the time we get to the 5th one, you will understand why the foods you eat in the morning specifically make a measurable difference to what happens in your retina for the rest of the day. But before we get to the foods, I need you to understand the scale of what we're talking about, because this isn't a small problem. According to data from the CDC's Vision and Eye Health Surveillance System, in 2019, an estimated 19.83 million Americans aged 40 and older were already living with some form of age-related macular degeneration. That number has almost certainly grown. Among Americans over the age of 65, a Harvard-based analysis of Medicare data published in 2025 found that the prevalence of AMD in that group was 10.4%, 1 in 10, and AMD is only one of the conditions that accelerates after 60. Cataracts, glaucoma, dry eye severe enough to impair vision. Each of these has its own biological pathway, and each of those pathways responds to the same fundamental nutritional deficiencies. Let me explain what those deficiencies look like inside the eye, and then I'll show you exactly how to address each one. The retina is not a flat surface. It has layers, and the most important of those layers for central vision is a region called the macula. The macula is roughly the size of a pencil eraser. It is responsible for everything you use to read, to recognize faces, to drive, to see color, to see fine detail. Inside the macula, there is a concentrated deposit of 2 pigments called lutein and zeaxanthin. These are carotenoids, plant-derived compounds that accumulate specifically in this region and function like internal sunglasses for your photoreceptors. They absorb blue-spectrum light before it can generate the oxidative damage that kills retinal cells. They quench free radicals. They protect the photoreceptors in the macula from the very process I described at the opening of this video. Here is the problem. Your body cannot make lutein or zeaxanthin, not a single molecule. Every bit of protective pigment in your macula came from your diet. And here is what the research tells us about how much people are actually getting. Studies using National Dietary Survey data have found that the average American adult consumes somewhere between 1 and 2 milligrams of lutein and zeaxanthin combined per day. But clinical studies measuring the amount needed to measurably increase macular pigment density, which is the actual measurable protective layer in your retina, show that you need somewhere between 5 and 20 milligrams per day for the effect to be statistically significant. Most people are getting less than a fifth of what their retinas need. A randomized, double-masked, placebo-controlled trial published in the literature on early AMD patients ages 50 to 79 compared supplementation of 10 milligrams of lutein per day, 20 milligrams per day, and a combined lutein and zeaxanthin formulation against placebo over 48 weeks. The group receiving 20 milligrams of lutein per day showed a statistically significant increase in macular pigment optical density, and here is the part that matters most. That increase in macular pigment density correlated directly with improvements in contrast sensitivity, which is your ability to distinguish objects from their backgrounds, the exact function that deteriorates first in early macular degeneration. This is not a fringe finding. This is a dose-response relationship measured in human beings using direct imaging of the retina over a controlled period. The macular pigment responded to the dietary intervention. So the first food on this list should be obvious now. Cooked kale. 1 cup of cooked kale provides between 23 and 26 milligrams of lutein and zeaxanthin. That is more than the 20 milligram dose that produced measurable improvements in macular pigment density in that clinical trial. Raw kale has slightly lower bioavailability because the cell walls and the leaf lock some of the carotenoids inside. Lightly steaming or sautéing breaks down those walls and releases the lutein into a form your gut can absorb far more efficiently. One more thing that most people don't know: lutein and zeaxanthin are fat-soluble. They require dietary fat to be absorbed into your bloodstream at all. Cooking kale in olive oil, or even eating it alongside a fat-containing food, increases the absorption of these carotenoids by a factor that researchers have measured as 3 to 5 times higher than eating it with no fat at all. If you eat steamed kale with water and nothing else, a significant portion of that lutein passes through you unused. Add olive oil. It changes the entire equation. Before I move to the second mechanism, I want to make sure you understand why the connection between lutein and macular pigment matters so specifically for the pattern of symptoms people over 60 tend to notice first. Because this is the part that makes people recognize what's happening in their own vision. The macula's central point, called the fovea, is where your highest resolution vision is located. It's the region you aim at when you read, when you thread a needle, when you look someone directly in the eye. The fovea has the highest density of cone photoreceptors in the entire retina. It also has the highest concentration of macular pigment. When macular pigment thins with age and dietary insufficiency, the foveal cone cells are the first to become vulnerable to oxidative damage. This is why the very first visual change people notice in early macular degeneration is typically not a blur in their peripheral field. It's a subtle shift in their central vision. Words on a page look slightly less crisp than they used to. Reading in dim light becomes more effortful than reading in bright light. Colors look slightly washed out compared to how they remember them. These are not the changes of aging lenses. These are early signatures of macular pigment thinning and the beginning of photoreceptor stress. And the majority of people dismiss them because they happen gradually, across years, and because they assume it's simply what happens when you get older. It is not simply what happens when you get older. It is what happens when your retina has been undernourished for years. Now, there is a second biological process happening in parallel that the lutein story doesn't address. And this one is less discussed, but it is directly relevant to why vision decline accelerates after 60 in a way that feels faster than it should. Your retina is one of the most vascularized tissues in your body. It is served by 2 separate blood supplies, one from the retinal blood vessels that run across its surface, one from the choroidal circulation underneath it, which feeds the photoreceptors and the retinal pigment epithelium from below. Both of these systems depend on microscopic vessels, many of them smaller than a human hair, to deliver oxygen and nutrients to the most metabolically demanding cells in your body. With age, these microvessels undergo structural changes. The walls thicken. The parasite cells that regulate blood flow through the capillaries begin to degenerate. Choroidal thickness, which can be measured directly using modern imaging, shows a consistent decline with age. What this means in practice is that the outer retina, where the photoreceptors live, receives progressively less oxygen and fewer nutrients as you get older. And when those photoreceptors are already under oxidative stress, reduced blood flow doesn't just slow their recovery, it accelerates their decline. This is where the second food becomes critical. And the evidence here comes from a different direction than the lutein research, but it is just as compelling. DHA, docosahexaenoic acid, is an omega-3 fatty acid that accounts for approximately 50 to 60% of the total fatty acid content in the outer segments of your photoreceptor cells. Those outer segments are constantly being shed and regenerated. They turn over roughly every 12 days. Every time a new outer segment is built, your body needs DHA to construct it. Without adequate DHA in the diet, the membranes of these outer segments become structurally compromised. Their ability to respond to light diminishes, and because DHA is also anti-inflammatory, inadequate levels allow the low-grade chronic inflammation that accelerates retinal vascular aging to go unchecked. A large prospective study in the UK Biobank, published in a major ophthalmology journal, followed 258 in 350 individuals. Over an average of 12.9 years, higher plasma levels of omega-3 fatty acids were associated with a meaningfully lower risk of being diagnosed with AMD. In a 2024 pilot study called MADEOS, a prospective, randomized, multi-center, double-blind, placebo-controlled trial published in Experimental Eye Research, found that patients with dry AMD receiving an EPA-rich omega-3 formulation showed a mean visual acuity improvement of 6 letters on the ETDRS chart over 24 weeks compared to placebo. 6 letters is clinically meaningful. That is the difference between reading a line on a vision chart and not. Now, I want to be precise here, because the evidence on omega-3 and eye disease is not uniform. The large AREDS 2 trial, run by the National Eye Institute, found that omega-3 supplementation added to the AREDS antioxidant formula did not further reduce AMD progression compared to the formula alone. What this tells us is not that omega-3 doesn't matter. It tells us that the benefit of omega-3 for the retina may be most relevant when someone is deficient in the first place, and when the formulation emphasizes EPA specifically over a generic fish oil blend. The MADEOS trial used an EPA-rich formulation specifically because EPA modulates the inflammatory pathways that damage the retinal vasculature. The practical application here is straightforward. Wild-caught salmon, sardines, and mackerel are the most concentrated dietary sources of DHA and EPA. A 3.5-ounce serving of wild salmon provides roughly 1,500 to 2,000 milligrams of combined EPA and DHA. 2 servings per week is the baseline. For people over 60 with measurable AMD, the research trend points toward higher daily intake, and an EPA-rich formulation is what the clinical evidence supports. What matters most is consistency over months, not a single meal. These fatty acids need to accumulate in your photoreceptor membranes before the structural benefit appears, and that process takes weeks. There is a third mechanism that we haven't talked about yet, and this one operates at the cellular level in a way that connects directly to the rate at which retinal damage accumulates. Your retinal cells, the photoreceptors and the RPE cells that support them, are among the most mitochondria-dense cells in your body. The reason is the same as the reason they are metabolically demanding. They need enormous amounts of energy to sustain the continuous phototransduction process, the molecular mechanism by which light is converted into an electrical signal your brain interprets as vision. Mitochondria are the organelles inside each cell that produce that energy, and with age, mitochondrial function in retinal cells declines. The mitochondria become less efficient. They generate more oxidative byproducts per unit of energy produced, and those byproducts directly damage the photoreceptors and the RPE cells they were supposed to be powering. This process is not unique to the eye. It is a universal feature of cellular aging. But in the retina, where energy demands are so high and the consequences of mitochondrial failure are so immediately visible, the impact is particularly significant. The compound that has the strongest published evidence for supporting mitochondrial function in retinal cells through dietary intake is one that most people associate with general antioxidant health: vitamin C. The concentration of vitamin C inside the aqueous humor of the human eye is 15 to 20 times higher than plasma levels. This is not a coincidence. Your eye actively pumps vitamin C in from the bloodstream against a concentration gradient, accumulating it at levels far above what circulates in your blood, because it is a critical antioxidant defense for the lens and anterior chamber. In the retina itself, vitamin C works alongside lutein and zeaxanthin to quench the free radicals generated by mitochondrial activity and light exposure. Large epidemiological studies have found an inverse correlation between dietary vitamin C intake and cataract risk, with the highest quintile of intake showing approximately 45% reduced risk of certain cataract types compared to the lowest quintile. And because the same oxidative pathways that drive cataract formation also contribute to retinal pigment epithelium dysfunction, the protective effects are not limited to the lens. This brings us to the third food, and it is one that many people consistently underestimate: bell peppers, specifically red or orange bell peppers. A single raw red bell pepper contains approximately 150 milligrams of vitamin C. That is more than a cup of orange juice, with none of the sugar spike. It also contains zeaxanthin, adding directly to the macular pigment pool, and it requires no cooking, no preparation beyond slicing. It can be eaten as the first thing on a plate every morning alongside eggs, which contribute their own supply of lutein and zeaxanthin in a highly bioavailable yolk-derived form. The fourth mechanism is one that ties everything else together, and most people, even those who eat well, are not thinking about it. Inflammation. The chronic, low-grade, persistent inflammation that increases with age is not just a vascular problem or a joint problem. It operates directly inside retinal tissue. Inflammatory cytokines, specifically molecules like interleukin-6 and tumor necrosis factor alpha, are measurably elevated in the retinal tissue of AMD patients compared to age-matched controls without AMD. These inflammatory signals accelerate the degeneration of the retinal pigment epithelium. They promote the growth of abnormal blood vessels in wet AMD, the form of the disease where fragile new vessels leak fluid into the retina and cause rapid central vision loss. And they degrade the tight junctions of the blood-retinal barrier, the selective membrane that keeps the retinal environment isolated from damaging substances circulating in the blood. The connection between systemic inflammation and retinal inflammation is bidirectional. What happens in your gut, in your fat tissue, in your blood vessels, eventually reaches your retina. And what happens in your retina contributes to the systemic inflammatory signal. People with elevated C-reactive protein, a standard blood marker of systemic inflammation, have higher rates of AMD progression. People with periodontal disease, another driver of systemic inflammation, have higher AMD prevalence. The dietary implication here is critical because the most potent anti-inflammatory food compounds we know of are not exotic. They are deeply common. Blueberries. 1 cup of fresh or frozen blueberries daily. The anthocyanins in blueberries, the compounds responsible for their deep blue color, are among the most powerful anti-inflammatory agents found in whole food. They directly suppress the NF-κB signaling pathway, one of the central molecular switches that turns on inflammatory gene expression in retinal and vascular tissue. Studies measuring C-reactive protein and inflammatory cytokine levels after anthocyanin supplementation have documented significant reductions. And anthocyanins from berries also cross the blood-retinal barrier and accumulate directly in retinal tissue, which means their effect is not just systemic, it is local, inside the eye itself. There is also a specific mechanism connecting anthocyanins to visual function that is worth understanding: the visual cycle. The process by which your photoreceptors regenerate the light-sensitive pigment rhodopsin after it is bleached by light requires continuous enzymatic activity. This process is directly impaired by oxidative stress and inflammation. Anthocyanins have been shown to support the rate of rhodopsin regeneration. That is why the earliest sign of dietary anthocyanin deficiency in visual function tends to be a slowing of dark adaptation, the ability of your eyes to adjust when you move from a bright environment to a dim one. People describe it as their eyes taking too long to adjust when they enter a dark room. That is not an age-related inevitability. That is a measurable functional consequence of insufficient anthocyanin intake, supported by the biology of the visual cycle. Eat 1 cup of blueberries in the morning. Frozen blueberries, picked and frozen at peak ripeness, have anthocyanin content comparable to fresh and are often more affordable. The benefit requires consistency. 1 cup once a week does not maintain the steady-state concentration of these compounds in retinal tissue. 1 cup daily does. Now, the fifth food is the one that surprised me most when I first encountered the evidence, and it connects to the blood supply problem we discussed earlier in a way that is both mechanically specific and actionable. Eggs, specifically whole eggs, eaten with the yolk intact. Here is why. The lutein and zeaxanthin in egg yolk are not more abundant than those in leafy greens. A large egg yolk provides roughly 200 to 300 micrograms of lutein and zeaxanthin, far less than a cup of kale. But the bioavailability of lutein from egg yolk is substantially higher than from plant sources. The reason is the fat matrix of the yolk itself. Lutein and zeaxanthin are fat-soluble, as I mentioned earlier. In plant foods, they are embedded in a cellular matrix that must be disrupted before they can be absorbed. In egg yolk, they are already in a lipid-rich environment that facilitates their transfer directly into the micelles that carry fat-soluble nutrients into the intestinal cells. Studies measuring serum lutein and zeaxanthin levels after identical intakes of lutein from eggs versus from leafy greens have consistently found higher blood concentrations from the egg source. When the same amount of lutein reaches your blood at higher concentrations, more of it reaches your macula. But there is a second reason eggs belong in this morning protocol, and it has nothing to do with lutein. Eggs are rich in choline, specifically in the form of phosphatidylcholine. Choline is a precursor to acetylcholine, a neurotransmitter that plays a role in visual signal processing in the retinal ganglion cells, the cells whose axons form the optic nerve. Adequate choline intake is associated with lower intraocular pressure in population data, and choline deficiency has been linked in animal models to degeneration of the retinal ganglion cell layer. This is the layer damaged in glaucoma. 2 whole eggs in the morning contributes meaningfully to daily choline intake. And when you add those eggs alongside sliced red bell pepper and blueberries, you have already addressed 4 of the 5 major mechanisms: lutein bioavailability, inflammation, vitamin C, and anthocyanin-dependent visual cycle support before you have left the house. Now let me be specific about what the research does and doesn't say. It does not say that any food or any combination of foods will restore vision that has already been lost to geographic atrophy, the late dry form of AMD, where retinal cells have died. Dead photoreceptors do not regenerate with diet. No amount of kale or eggs reverses that. What the evidence clearly supports is this: adequate intake of lutein, zeaxanthin, omega-3 fatty acids, vitamin C, and anthocyanins supports the biological mechanisms that protect retinal cells from the oxidative damage, the vascular insufficiency, and the inflammation that drive them toward that irreversible endpoint. The protective window is before the damage becomes structural, and for most people over 60, that window is not closed. Let me give you a practical framework. In the mornings, 2 whole eggs scrambled or cooked in a small amount of olive oil alongside 1 sliced red bell pepper and 1 cup of blueberries, fresh or frozen. That combination covers lutein via the yolk, zeaxanthin from both the yolk and the pepper, vitamin C from the pepper at more than 150 milligrams per serving, and anthocyanins from the blueberries at levels that research associates with measurable anti-inflammatory effect in vascular and retinal tissue. 2 to 3 times per week, include a cup of cooked kale or spinach with your evening meal, sautéed in olive oil, not boiled. Boiling leaches carotenoids out of the leaves and into the water you discard. The fat-soluble carotenoids stay in the leaf when you sauté them, and the oil increases absorption. 2 to 3 times per week include wild-caught fatty fish, salmon, sardines, or mackerel. A 3 to 4 ounce serving provides the EPA and DHA your photoreceptor membranes need. If you genuinely cannot eat fish, an algae-based omega-3 supplement that provides EPA and DHA directly is the appropriate substitute. Algae is where fish get their omega-3s to begin with. Algae-derived DHA and EPA bypass the fish entirely. A word on timing. There is a biological reason to consume these foods in the morning and early afternoon, rather than in a single large meal at dinner. The cells that transport carotenoids and fatty acids from your intestine into your bloodstream are subject to circadian influence. Absorption of fat-soluble nutrients, including lutein, zeaxanthin, and omega-3 fatty acids, is generally higher earlier in the day when digestive enzyme activity and bile secretion are at their daily peak. Your eye uses the nighttime hours for some of its repair and renewal processes. Giving it the nutritional building blocks earlier in the day means those nutrients have time to circulate and reach retinal tissue before the repair window begins. What should you expect from this kind of protocol, and when? In the first 2 to 4 weeks, you may notice subtle changes in visual comfort, specifically a reduction in light sensitivity and glare discomfort, which reflects the earliest functional changes associated with increasing macular pigment density. These changes are real, they are measurable with the right instruments, and they precede any change in measured visual acuity. Between weeks 4 and 12, if you are consuming adequate lutein and zeaxanthin consistently, studies show that macular pigment optical density measurably increases in this time frame in most people who were deficient to begin with. That thicker pigment layer provides a more effective filter against the blue light that drives photoreceptor oxidative damage. If you are also maintaining adequate omega-3 intake, the inflammatory markers associated with retinal vascular aging begin to move in a more favorable direction over this same period. Beyond 3 months, the cumulative effects of consistent anti-inflammatory and antioxidant nutritional support begin to influence the rate of progression of early AMD in people who already have early changes. This is the biology behind what the AREDS and AREDS-2 studies, the largest clinical trials ever run on AMD and nutrition, were designed to measure. The AREDS study found that people with intermediate AMD who received a specific combination of antioxidants, including vitamin C, vitamin E, beta-carotene, and zinc, reduced their risk of progressing to advanced AMD by approximately 25%. AREDS-2 subsequently found that replacing beta-carotene with lutein and zeaxanthin produced outcomes that were at least as good and were safer for the large proportion of study participants who were current or former smokers. These are not small numbers. A 25% reduction in the risk of progression to the form of AMD that causes irreversible central vision loss is clinically significant for any individual at risk, and most people over 65 are at some level of risk, because the prevalence of at least early AMD in Americans 65 and older is 1 in 10, and most of them don't know it until someone looks at the back of their eye with the right equipment. Vision is not a sense that most people think about protecting until they notice it failing. But the biology doesn't work that way. The decline begins years before the symptom. The nutritional window for support is open during those years, and the foods that support retinal health are not expensive, not exotic, and not complicated to include in a morning routine. Vision is not a sense that most people think about protecting until they notice it failing, but the biology doesn't work that way. The decline begins years before the symptom. The nutritional window for support is open during those years, and the foods that support retinal health are not expensive, not exotic, and not complicated to include in a morning routine. Let me say something directly about what vision loss actually means in practical terms for life after 65, because I find that people don't fully connect the biology to the stakes until someone puts it plainly. Vision is the sense most directly tied to independence. It determines whether you can drive, whether you can read your own medication labels, whether you can safely navigate stairs, whether you can recognize the people you love across a room. Studies on falls in older adults consistently find that visual impairment is one of the strongest modifiable predictors of fall risk. People with moderate to severe vision impairment are 3 times more likely to experience a fall that requires medical attention than those with intact vision. Falls in people over 70 are not minor inconveniences. They are a leading cause of hospitalization, of nursing home admission, and of death. Vision does not exist in isolation from the rest of your health. It is connected to your mobility, your independence, your cognition, and your social engagement. AMD specifically damages central vision, which is the vision you use for almost everything you care about doing. Peripheral vision, which AMD tends to spare in the early stages, allows you to walk around, but it does not allow you to read. It does not allow you to recognize a face. And the research on quality of life in people with advanced central vision loss is consistent. The emotional and cognitive impact is profound. Depression rates in people with vision-threatening AMD are approximately 3 times higher than in age-matched controls with healthy vision, not because vision loss causes depression on its own, but because vision loss restricts the activities that give daily life meaning. None of this needs to be inevitable. The biology is not a fixed sentence. Eggs, blueberries, red bell pepper, cooked leafy greens with olive oil, and fatty fish 2 to 3 times a week. That protocol covers the mechanisms that matter: macular pigment density, retinal anti-inflammatory defense, photoreceptor membrane integrity, mitochondrial protection through antioxidant support, and the visual cycle function that determines how quickly your eyes adapt and how clearly you see under changing light conditions. These are the structures your eyes depend on, and they depend on what you eat. If you found this useful, the next video covers the second half of this picture, the specific daily habits that drive retinal oxidative damage faster than age alone, and most of them are things that people over 60 are doing every single day without realizing the cost to their vision. I'll see you there.